So while we're getting people up here, Allison, so it comes up a lot about, how many, say, Chiari families do we need to make some headway in this? What if you had your dream, if we can round up around the world, these are, say, kids or adult, as Dr. Keating said, these are different maybe. But let's just say kids, Chiari families, how many family members are important to you and how many total would be helpful?
Oh, is it on? I thought I turned it on. Can you guys hear me?
Yeah.
So, my dream would be thousands of patients and thousands of families, there's still some work to do, but I think, a number of us kind of across the U.S. in particular, at least have a few hundred. So I actually think it might not take too much more work to really get to reasonable numbers.
It's good to know. Questions from the audience. There's a lot of different kinds of things we went over in this session, so I'm sure there's a lot of different interest of what people have to say. So we'll try to connect them any way as we can. So
everyone was wonderful. Thank you so much. Regarding the sample sizes that you're speaking of and speaking to all of you, are there studies that you could contribute to, say, like a meta-analysis, if you do have raw DNA that's from, other, like a company, but that would be safe for your privacy, because a lot of that is controversial. I did my sequencing, the whole thing, because I wasn't going to pay for it one by one, and I would love to contribute. So with that, and then also because of genetics, but also a lot of you are involved in studying different metabolizings of medical treatments, even the cannabinoid system. I'm curious, and if anyone is collaborating with epigenetics of like nutrigenomics, even how that is affecting mast cells, like how you metabolize, because my understanding of genes is very naive, but they work in unison and they are not consistently, even in the cycles of life. You have different stages where cortisol changes, obviously estrogen, progesterone. I was misdiagnosed with Addison's disease before I got into all of this. So I wonder if there is a chronic impact of having these conditions, chronic stress, that sort of thing, just our different societies, the global changes going on, if that is in fact impacting how our genes, whether they're a genetic snippet there, or not, but how it's expressing.
Yeah, those are great questions. And you sound like you're very informed, actually. Yes, I think, in terms of contributing sequence data, if you have it, we would, have to work through IRB and getting the data from you. But, absolutely, that could be contributed. So, that's a really interesting idea, because I hadn't really thought about that there may very well be many patients out there who have had sequencing performed on a clinical basis, that those data exist, and we would just have to pull them together. So that would be great. I'll touch quickly on epigenetics, because I think maybe, Ian, you might be one to address that a little bit. So I do epigenetic analyses. I haven't for Chiari, but epigenetics is really fascinating. And so just quickly for the whole audience. So, we talk about our sequence, but, on each of, our A, C, T, G, on the C's, we can have, I'll call it a little hat, we'll say, that's a methylation hat that sort of sits on it. And when you have a lot of C's in a row and they've all got hats on, that tends to turn the gene off. So you're absolutely right. You know, epigenetics is one of those ways that we sort of fine tune, if you will, whether it's developmentally or in response to nutrition or the environment. This is how our bodies kind of try to fine tune things. So that would be very exciting to look at.
Dr. Maitland, you want to comment on sort of daily stress and what, epigenetics in any way? Sorry, you're muted in some way. Maybe it's us.
I'm good. Can you
Okay,
hear me?
now we're better. Yep, thanks.
Sorry, I'll turn it up. So I was saying that at Metrodora, we actually have an IRB approved protocol where we are actually doing whole genome sequencing with families that are enrolling into the program. And what we're hoping to do is not only, so we're taking signs and symptoms, pairing it with the variants that have been identified, and then we've partnered with a AI company called Precision Life, where we're starting to not only chart out the pathways that are being more actively engaged, but also to identify therapies that either repurposing old therapies or seeing what's in the pipeline, as well as pairing it with physical medical therapies as well. I have to tell you, the immune system has been kind of redefined within the past decade. Before, it was the innate immune system and the adaptive immune system. And we know that the adaptive immune system will modify just so you can have such a very large tailored repertoire of both the T cell compartment and the B cell compartment. But the part of the tissue defense that is the most intriguing at this point in time are can only really be defined with genetic testing. And that's the hyperinflammatory or the autoinflammatory syndromes. And we're starting to see, I guess what's really interesting about being in Salt Lake City as a New Yorker, as a scientist, I was drooling over Amish studies, but I have to tell you, Salt Lake City, the genetic pool there, the Mormon genetic pool there is actually pretty impressive. And we've unmasked several autoinflammatory syndromes that have been identified just with genetic testing, and then we're able to tailor therapies towards the variants that have been identified. So unfortunately, our genes have been under assault, I would say, for the past 50 years, and selecting out for gene products that are able to deal with the environment, but not necessarily in a constructive fashion. I would have to say, for instance, one gene that is actually quite common in the Caucasian population in the mast cell compartment, which will not be picked up by whole genome sequencing because it's actually a multiplication of the wild type gene, is hyperalpha-tryptosemia, which has been shown to impact about 7% of the Caucasian population. And we see that there's variable penetration, even though it's an autosomal dominant inherited gene. So you're talking about what the environment that instructs the mast cells to do, and then how the mast cell adapts its machinery to deal with the microenvironment that it's taught to do surveillance and then execute certain chemical mediator release patterns. So I have to tell you, the question you ask is a torch that's lighting the way to try to understand how our bodies are negotiating an environment that has been completely revolutionized in less than 50 years.
Very interesting. No, thank you. Great question. You see how it spurred a lot of really great discussion for that. Other questions, Dr. Martin?
Yes, I have a question for Dr. Maitland. I'm intrigued by the relationship between mast cell activation and hypermobility. And it seems to me, having read the initial paper on alpha tryptasemia, they report both, joint hypermobility, and they also report migraine headaches. And I'm a headache doctor, so that interests me as well, that maybe that could suggest mast cell involvement, not only in hypermobility, but also in migraine headaches. So I wonder whether that's not your best evidence that's involved, but I'm also intrigued by the possibility that if mast cells can be activated by the environment, which obviously they can, if there's a relationship between mast cells and joint hypermobility, could it be acquired? And
Yes.
you think it can? Do you have evidence? And then I also want to know what specific pathophysiologic way do mast cells actually lead to hypermobility?
So the best information we have regarding the relationship between mast cells and actually there's two. One regarding primary mast cell disease, and that's the hyperalpha tryptophanemia study. But the acquired is the secondary mast cell activation disease, meaning there's nothing wrong with the mast cells, but there's something wrong with major triggers of the mast cells, including going through the IgE receptor, going through the complement receptor, and the fact that complement mutations have been identified, both hereditary and secondary angioedema, going through the complement pathways, which will modify mast cell activation. Sometimes it's a full-blown episode, sometimes not. But what I find to be very interesting is that having a increase of an enzyme that can clip collagen, can clip a lot of potent chemical mediators, that the individuals that are diagnosed with hyperalpha-tryptophanemia have a higher level of this enzyme floating around that's active, which can modify the connective tissue. But we don't see it in all the patients that inherited that mutation, nor do we see a dose response, because we've seen duplication, triplication, and quadruplication of this wild-type tryptase gene. But the quantity does not correlate with the penetration of the disease. So we have seen family members that have 4 copies. I mean, it's like going from a Saturday night special to like an AR-15. Some of the family members are like, I have a little bit of this, a little bit of that, but I'm not as bad as John who brought us here. So that goes to show you that the mast cell is under control of the surrounding microenvironment, meaning the signals that are stimulating the receptors that then lead to different pathways of chemical mediator release. So that being said, we have seen individuals that have inherited more than one mutation. And depending on their exposures, those other comorbid disorders can then tickle the mast cell compartment to be at a higher or lower activity. And I can even tell you, even with systemic mastocytosis in children, they have a mutation in the major growth factor of the c-KIT gene. But 75% of these children, if they show manifestations of the cutaneous mastocytosis before the age of 6, will outgrow the mutation. Like they completely start regressing on their own. The cutaneous lesions completely regress. So leaning into central nervous system manifestations, it all depends on where these mast cells are misbehaving and what are they releasing. And so I thought it very interesting that, and I don't know if Dr. Klinger will address this, but she looked at individuals that had to undergo revisions of their occult tethered cord surgery, and she saw an increased presence of the mast cells in an activated form in the tissue that she biopsied. And she actually correlated that with the tensile strength of the ligaments that were removed from the surgical specimens. So I think we need to talk about what is the burden of mast cell activation, whether it's increased number or increased chemical mediator release, and then what's the target? Because you can definitely modify just with growth factors and also the ability to modify blood flow. And you want to talk about migraines, like I'm absolutely fascinated on how well people are doing with those anti-CGRP medications. We're actually using them for helping with desensitization as well. So it keeps them quiet, mast cells quiet. So, and also you think about tricyclic agents again with migraines, that's H1, H2, H3. So it depends on how you can keep those mast cells quiet, and then also potentially modifying what they're releasing and when they're releasing so that they don't adversely affect the metabolism itself. I'm sorry, it's a little bit, all of this has just been coming together within the past 2 years. trying to put together this crosstalk between the nervous system and the mast cell compartment, both in the central nerve, both in the brain, the spinal cord, but also in the periphery as well. And I have to tell you, when it comes to mast cell dysregulation, really the three top complaints, whether you're talking about primary mast cell disease, such as systemic mastocytosis or hyperalpha-tryptosemia, or you're talking about secondary mast cell activation disease, meaning not an increased burden, but just increased burden of chemical mediator release. The three major complaints is gut, respiratory, and neuropsychiatric, including headache disorders, mood disorders. And so I think this is going to be a robust area of research just because we're thinking about the crosstalk between these three key organ systems that make up every part of one's body.
I'm curious, Dr. Gray was talking a lot about CSF and myelograms and pressure. I don't know how often you check the CSF. We often do basic tests. But do you think there's any value in looking at the CSF from these folks? Or can you pick up everything you need in the blood?
What would you want?
Mast cells are tissue-based cells.
What would you want to check?
Well, with Dr. I have to tell you the effort by Dr. Klinger regarding the phylum came out of an ASAP meeting almost 9 years ago, where I'm like, you need to stain for mast cells because if you use an H&E stain, you won't see them. And not only should you, and this is a tissue-based cell. They're supposed to restrict their activity to the area where they're guarding. So I would stain for mast cells, and then I would also stain for whether or not they have upregulation, and then also I would look for what chemical mediators are de novo synthesis in these.
Okay. I can, I have to talk to the lab and see how I can order a mast cell evaluation of the CSF because right now I only have 4 things: cells, protein, glucose, and some other things. Right, no, we're just so
primitive in what we test for, but maybe there's opportunity.
Yeah, I'll look into looking for mast cells.
Yeah, that's no different in the Chiari we call failures. We look at all the clear things that what we would do is it's still crowded, instability, pressure, but we're missing something. And this is the bucket we miss because we don't know how to test for it.
Yeah, so I'll check it out.
Honestly, the frightening thing about mast cells is they were one of the first immune cells identified back in the 1800s, and they didn't got a job until 1989. So it's sad, and then, and what's really sad is how everybody starts blaming mast cells. I'm like, you really not a single human alive that lacks mast cells, right?
Other questions out there? Yeah, here's a question over here.
Hi. I think I heard somebody else reference the Norris lab, and I know they also had a paper in addition to the specific kallikrein gene that was looking at just clusters of their patients within their study. And I'm just curious about the thoughts of the cluster 3, which seems very relevant for this population. And specifically, it had a high prevalence of Chiari and tethered cord versus the cluster 1, which looked very similar in a lot of ways, but did not have Chiari and tethered cord. My daughter very much fits in that third cluster, and I just don't know if that is pointing toward breaking down the broad EDS, hypermobile EDS, into subsets that would have different presentations. If any of those genes that Dr. Ashley-Koch mentioned, or I'm very curious if this is just how people present and not really related to the actual genetic studies, or what to do if you see this cluster and your child fits in it. Like, does it inform treatment or how to approach things?
Well, I will speak just to the genetics and, the data that I showed you that our group and some other groups have done where we've sort of taken the clinical presentation and sort of stratified people based on that. That really has helped us find the genes. in EDS is difficult because there are lots of genes, and even in our analysis, I mean, we had signal in almost every collagen gene that we, tested. So, I think, it can be difficult to sometimes say, well, this is definitely going to help us. But I do think if, we can use the knowledge that all the clinicians and the patients and what your experiences and the symptoms are to help us sort of pull people into different related groups, I think that will ultimately help.
Cormac and Vez, I mean, you have pediatric and adult Chiari centers represented. How do you incorporate genetics into your center practice? Who do you decide to order genetics consult on? Are we missing the boat? Should we do it on everyone? Should we just then give us your thoughts of how you? I know we send to genetics and they say that can't help us. They often don't like these consults, but it's a culture thing too. But I'm just curious.
Yeah, I'll be the sort of the cynic, I guess, and it's mostly based on the data that I showed earlier. I don't think there's going to be a simple genetic explanation with Chiari. I think it's going to be a very complicated one, multifactorial, and it certainly does run in families. We know, as Dr. Ashley-Koch eloquently showed us, and there's a lot of reasons for thinking that there will be some genetic explanations. But again, I think it's going to defy a simple genetic explanation. There's just no way to explain the population distribution on a simple genetic basis. The way I sort of think about this, and it's probably a professional trained geneticist is probably going to be aghast when I say that, but I've always thought of Chiari in a genetic sense as something like facial structure almost, where you can go to a family reunion and you can look across the room and say, oh yeah, that must be a relative of my Uncle John because he looks exactly like my Uncle John. But that's a very complicated genetic outcome to have that facial structure, right? And I feel like Chiari is the way I understand it. None of us really know, frankly, but the way I've always understood the genetics is probably it's going to be genetic on some sort of a level like that, where we might find, there's not going to be one gene, but we might find 1,000 genes that are contributory in various ways and in various levels in different people. Anyway, with that as background, I would say a small minority of my patients are actually seen by genetics because I've found that, as you have pointed out, they're not getting much clinical satisfaction from those consults in general. The exceptions are going to be people who have other signs of genetic syndromes going on beyond just the Chiari malformation.
Syndromic of some sort. Yes.
Yeah, I would agree. I mean, sometimes patients will ask for it, and I think I completely agree with everything that Cormac just said, and I think the sense is what information is going to be at hand that's going to change what I do to take care of this patient. Someone with EDS has some other type of familial, lineage of absolutely it's helpful, but I mean, in my experience from the adults, it's more of an educational as opposed to is it going to change ultimately what we do. And I would also agree. I think Chiari is clearly multifactorial. I don't think there's going to be one gene that's responsible for this, or even a cluster.
Yeah, these are just such tough studies to do. As you probably know, around the country and the world, there are groups that have formed. Chris Colley's lab, for example, looking at hydrocephalus and pulling in samples of hydrocephalus. Dave Limbrick's doing some of this. Arachnoid cysts, a lot of people are sending in, the blood or for analysis of they're doing the whole genome sequencing and everything. These are big studies, but it's always back to the phenotype. Well, are all these the same kind of people with arachnoid cysts? Hydrocephalus, is it slit ventricle, pseudotumor, congenital, secondary hydrocephalus? There's so many different bags. Look at Chiari. We would need the films. We would need quite a bit of data to show Allison, that these are the same phenotype of people. We probably age But there's a lot of interest in doing this because of you all. You all have expressed that interest that we need to do more, willingness to send to some repository to start looking at these connections. We can't do it without you. And with social media now, there's a much greater opportunity to pull these people in. But then it gets very challenging if you can't send the pictures. Do they ever have a Chiari in the first place? That's not so simple, as we've been talking about all day. So these are really tough studies to do. And CSF, we should start looking at more things. And Allison, remember, we looked at these with what is the right thing to test? It's not just blood. Remember, we looked at Durham.
That's right.
Around the Chiari, thinking Durham is going to be the answer. Is it muscle? Is it bone? Looking at the skeleton.
I like the CSF idea.
Yeah, we're back to Linda's team. So that's why Linda's not retiring. She promised 10 more years today. Yeah.
An
To Keating.
LP clinic.
And this is Bob Keating. Great talk as usual. You alluded to this in one of your slides about the veterinarian stuff. We know that the whole neuroimmunological pathology has exploded in the last decade or more. It's the same thing in the pet world, and dogs and cats are living shorter, same issues. Any thoughts on what's the shared etiology about that?
So I believe they're being subjected to the same environmental exposures that we are. And, very similar genes, especially on the level of the epithelial barrier. These alarmins, if you look at how asthma and atopic dermatitis has moved to let's restore the barrier. So you're less likely to end up with leakage of the barriers that are allowing these antigens to come in and engage both the somatosensory network and the mast cell compartment. So if you help restore the barrier, reduce those alarmins that are recruiting in that neuroimmune network, we're starting to see better control of these immediate hypersensitivity disorders. And if you can control those immediate hypersensitivity disorders, you can then reduce the risk of it being recruiting in a more permanent form of the adaptive immune response. And I find it very interesting that we're seeing, like, for instance, the major genetic variant that's associated with atopic dermatitis is not a marker in the classic innate immune system or the adaptive immune system. It's a structural protein of the skin. And we're seeing, and I think I'm not seeing as much literature in the respiratory epithelium, but it's amazing that we're finding issues with the alarmins that recruit in that sensory neuroimmune network. And also we're starting to see the chronic injury of the borders and the proteins in there is allowing for the development of autoimmune disorders. I have to tell you, I've never seen so many individuals have acquired angioedema. Just like we're starting to see autoantibodies against the mast cells and the basophils, we're starting to see autoantibodies against components of the complement system. And that's not a good combination, because then you can end up you're talking about letting the hounds out. Activating the complement system without regulation is a recipe for disaster. And so I think, between, and I'll have to send you some of the earlier work that I had seen about mast cells in some of the breeds of the dogs. Like, they're the ones who are an increased risk for mast cell tumors. I'm like, well, I mean. So I really think it's a question of, the living environments that we're in that's causing chronic irritation and epithelial border damage. And I think that's recruiting in the neuroimmune network, causing epigenetic changes on how do you deal with, for instance, these forever chemicals that are able to penetrate through the epithelial barrier and directly engage the nerves as well as the mast cell compartment. We're so chronically exposed to things that are just stripping our barriers and activating that network. And once that network is out of it, unless we turn it down, we've completely seen a change in the burden of disease in less than a century, from infectious to immediate and delayed hypersensitivity disorders. I've never seen so many advertisements on TV for medications to kind of shut down your immune system.
Interesting. Thank you. Last question.
Question for Dr. Maitland. I was just curious if a patient does need to go down the route of finding out if they have a connective tissue disorder, where should they start? What, a doctor such as yourself, rheumatologist, a different physician, what types of test and blood work should they make sure they ask for? And then I'm just curious in particular if there are any other connective tissue disorders that affect vision specifically, maybe due to inflammation, just any that we haven't touched on. I know, EDS is most popular in POTS, but maybe if there's some others that we haven't heard of.
I've seen it in osteogenesis imperfecta. I've seen it in Marfan's. So you're really talking about global connective tissue, increased susceptibility to tissue injury. So to answer your first question, I would strongly recommend interacting with the patients, patient societies, Ehlers, Mast Cell Disease Society, Ehlers-Danlos Society, Dysautonomia International, all these practitioners that have been entrenched in this space and haven't run away in the past decade. That's one. Regarding testing, believe it or not, HEDS has a formal checklist for screening. And then there's a robust INVITE panel, or you can do whole genome sequencing that's been identifying more variants. But I have to tell you, we're starting to, in many ways, I think some forms of hypermobile Ehlers-Danlos syndrome, I think that's acquired. And I think it's because we've identified at least 2 variants. Sorry. I think they've been at least 2 variants that are identifying issues in the complement system that has the ability to cause a global connective tissue issue. So the kallikrein system that Dr. Norris and Dr. Gessmer have identified, and then also periodontal Ehlers-Danlos syndrome, which is really interesting. These individuals are actually at increased risk for vasculopathies, including both distal and also impacting the cerebral vasculature as well. So I think we're moving to a world of impaired tissue defense and impaired tissue repair following injury that may be explaining the surge of immediate and delayed hypersensitivity disorders that we're seeing.
Well, thank you all. Please thank our panelists. Really great session. So, thank you.